Well, thanks everybody for your patience. I know this is the last presentation of the afternoon, so bear with me. This is going to be science, a more science-focused presentation. So before I start, I would like to thank Lydia and the other organizers of the conference for really the privilege to speak to you this afternoon. I've learned a huge amount today. And so we're always learning in research. I'll make two disclaimers. So I'm clearly not a clinician. I'm not going to speak about clinical aspects of urology. You've just heard a lot about that from Drs. Tan and Elder. The second disclaimer is I really have yet to work on the ACTA2 mutation. Dr. Mussolino and I are in the very embryonic stages of a collaboration, shall we say. But what I am going to do is tell you a story about smooth muscle regulation, and a way in which it might be relevant to some of the pathology that those afflicted with the ACTA2 mutation are experiencing. So let's get into the science. So before I begin, I do want to acknowledge two key collaborators. I'm going to share some published work. And the two principal collaborators are Diane Bielenberg, who first introduced me to the protein family I'm going to talk about. She's actually a cancer biologist at Boston Children's. And the second key collaborator is Mary Rose Sullivan, who is really an expert physiologist. She works at the Boston VA healthcare system in West Roxbury. And really, it's the three of us together have developed this project over the last decade or so. I'll note some other people as we go along and obviously acknowledge support from federal funding agencies. So you heard a little bit about the pathophysiology as it pertains to the bladder in MSMDS. And what I want to focus on is this issue of the hypotonic bladder, a structure with reduced contractility. And in particular, I'm going to focus on smooth muscle, which is appropriate for today's discussion. Having said that, there are probably also aspects of innervation and the vasculature that may be relevant to the story I'm going to tell you. So just to begin, I want to present a really simplistic view of the response of smooth muscle to various types of damage. So we have a hollow organ, we have a lumen lined with epithelium, surrounded by one or more layers of smooth muscle. And in response to a variety of insults or injuries, such as obstruction, aberrant innervation, infection and inflammation, or indeed the ACTA2 mutations that we've been discussing today, the hollow organ can undergo a spectrum of changes, but you can imagine two broad states, both characterized by inappropriate function of smooth muscle cells, dedifferentiation, proliferation, potentially death of smooth muscle cells. And what that will result in, whether we have enlargement or reduction of the smooth muscle compartment, is a loss of normal contractility. And in the case of this presentation, we're talking about urinary dysfunction. I will say that we've studied gut smooth muscle as well, and certainly see some parallels with the work that Dr. Rahman presented earlier this morning. So just to give you a sense of how we approach some of these studies in the lab, I want to share four broad ways that we can study bladder function and smooth muscle cell function. So we can perform cystometry or urodynamics in experimental animals, much as it would be done in humans. And that's shown just in this simple schematic here. So essentially we can implant a catheter in the bladder and monitor its behavior upon filling. We can record urine output and the pressure in the bladder as it fills. So this is applied to obviously living, awake animals. Another type of assay that we can do is called the void spot assay. This is incredibly simple. So you essentially put a mouse in a cage with filter paper lining the bottom of the cage, and it turns out that urine will fluoresce under ultraviolet light. So we can actually get an example of voiding behavior based on the pattern of urine spots. Dr. Mussolino's lab has used this assay and has applied it to their ACTA2 knock-in mouse model. And what it allows is a non-invasive evaluation of bladder function over time. I will say there are some caveats with this, since mice and other animals use urination as a way to mark territory. So it can often be an assay that goes far beyond urinary tract function, where an animal is establishing dominance, for example. But it is a very useful way to monitor the consequence of an insult or an injury to an animal over time. When we think about analyzing smooth muscle tissue function or cellular function, we use one principal technique, and that is tension testing. You heard a little bit about this again this morning from Dr. Rahman. Here we can take a bladder, we can divide it into strips, and we typically remove the lining cells, the urothelium. We can then suspend these in an organ bath and place the tissue strip under tension, and then stimulate it with both soluble factors added to the organ bath, or we can stimulate it electrically and record the response that is evoked by those stimuli. And then we can turn this information into quantitative assessments of muscle contractility. I'm a cell biologist and biochemist by training, and so what I like to study really are cells, and we can study the contractile activity of individual cells or cells in a group using a technique called traction force imaging. And what you're seeing here is one cell that is sitting on a sticky substrate, and the hotspots that you can see on this image here and here and here, those represent the cell exerting tension on the underlying substrate. So we can actually quantify the extent of contractility. And then another interesting technique, we haven't used this very much, it's complicated, but we can do something with a long and complicated name called optical magnetic twisting cytometry. Essentially, this allows you to measure the stiffness of a cell. And this is directly relevant to the cytoskeleton, in other words, the cellular skeleton, which is largely formed by actin. And so in this technique, we can attach magnetic beads to the surface of the cell. We can put the cells into a magnetic field, apply the magnet, and essentially twist this bead. And the extent to which the cell can withstand that twisting is a measure of cell stiffness. And so we have used various combinations of these techniques, both in whole animals, in tissues, and in cells, to investigate the behavior and the relevance of a particular protein family, which I'm going to introduce in the next few slides. So this family, they're called neuropilins, and there are two such members. So these are proteins that sit on the surface of the cell, and they essentially act as co-receptors for two broad groups of signaling receptors. So the first is the vascular endothelial growth factor receptor, or VEGF receptor. And again, the names here don't really matter. It's just that I'll distill this into the take-home points when we get to the end of the slide. But the VEGF receptor essentially controls vasculogenesis, so the creation of vessels, and a process called angiogenesis, which is also the creation of new vessels. There's another class of surface receptors called the plexins. And again, these are transmembrane proteins, and they transmit particular kinds of signals into cells. So the neuropilins will bind other classes of molecules, and these are soluble factors, in this case, a factor called VEGF. And so neuropilin, VEGF, and VEGF receptor will form a signaling complex, and that will transmit information into the cell to promote vessel growth. Another class of soluble factors are these things called the class 3 semaphorins. And as the name suggests, these send signals to other cells. And this really has been very heavily studied in the nervous system. So semaphorins and neuropilins and plexins are instrumental in axon guidance during development. So they guide axons to their terminal destinations to set up essentially the nervous system. What our group found, this was in 2007, 2008, was that the neuropilins were very heavily expressed on smooth muscle. So this is an observation that had been made, but not really commented on. And we proceeded to pursue this in our group. I'm just going to give one other bit of science information, and please don't freak out when you see this slide. So this is this branch of the neuropilin 2 signaling activity that we care about. So neuropilin 2, SEMA3, and plexin. So these are sitting on the cell membrane. And what we care about in terms of cellular function and smooth muscle cell function is the following. So when these three proteins come together on the cell surface, there's a series of signals that is transmitted into the cell. And again, don't worry about the names, but through a series of phosphorylation events, these are activating events. What happens is we turn this key regulator, which is called RhoA. This exists in two different states. One is active, one is inactive, and this signal turns Rho off. As a consequence, a partner for RhoA called ROC is also turned off, and through a series, a cascade of events results in the generation of the soluble form of actin, G-actin, that's globular actin, that is consistent with cellular relaxation. When Rho is active, there's a different series of events that is occurring that results in the filamentous form, or F-actin. So that is the structural constituent of the actin cytoskeleton. And when we're in this state, together with myosin, which is the other component of smooth muscle contraction, we actually have contraction. So this complex here, the state of Rho and its partner ROC dictates whether the cell is relaxed or contracted. And this is regulated by this protein, neuropilin 2, in conjunction with the other members on the cell surface. So why did we study neuropilin 2? Well, my colleague Diane Bielenberg came to me one day in 2007 or 2008 with an image of a mouse pup, actually. So this is a sagittal section, a section through a mouse pup, and it is a mouse strain. Pardon me, okay. that allows us to see where neuropilin-2 is normally expressed based on a blue color that comes from this constituent LAC-Z. So you can see some blue color in the brain, you can see some blue color down the spine. But what really had caught her eye was all of this blue color in the abdomen of this mouse. And the structure marked with the asterisk is the bladder. And we also see a lot of blue signal in the surrounding structures. These are the sections through the gut as they move back and forth the abdomen of the mouse. So as I say, this observation had been made. People had found neuropilin-2 in the gut and the bladder, but hadn't really pursued it. That's not at all surprising to me because people don't really study the bladder very much. But this was an interesting observation. When we compare the distribution of neuropilin-2 with the distribution for alpha smooth muscle actin, you can see that they are largely overlapping based on these serial images here. So we went on to look at this further, and this is the bladder shown at much higher magnification. And you can see when we blow up this particular region, all of the blue signal is within the smooth muscle. And again, we have an image stained with alpha smooth muscle actin that shows a very similar distribution pattern. So this really verified that neuropilin-2 was highly enriched in smooth muscle. What's also interesting, when we look in the vessels, and here we're fortunate to have an artery and a vein next to each other, the blue signal is actually restricted to the lining cells of the vessels. Neuropilin-2 does not appear to be expressed in vascular smooth muscle. And at least otherwise healthy animals, we don't see neuropilin-2 in vascular smooth muscle. There is some evidence that the expression will turn on when the vessel is injured. And so that may be relevant to the ACTA2 scenario. This is heavy stuff. This is my third microphone. Maybe I'm getting a hint. So I think this is certainly an interesting observation, and it may well be relevant as we go through the talk. So this is how we got into this study. Neuropilin-2, very highly enriched in the bladder wall. What is it doing there? So we did some fundamental cell biology. Here we're just showing expression of neuropilin-2 in human bladder and colonic smooth muscle cells. Consistent with the picture I've just shown you, we don't really see as high expression in cells of vascular origins. So these are umbilical artery, which is an unusual smooth muscle line, but pulmonary artery, coronary artery, we really don't see as much signal. So that's consistent with the picture that we see in tissue. This is what human bladder smooth muscle cells look like in a culture dish. And I mentioned that neuropilin-2 binds this protein, one of these SEMA3 proteins, and in particular, a protein called SEMA3F. So when we add SEMA3F, the cells change shape, and this is consistent with that cell relaxation branch of the signaling pathway I just showed you. If we inhibit Rho A, and I suggested that when this complex forms on the plasma membrane, that signal that transmits down into the cell is inhibiting Rho A. So if we inhibit Rho A chemically, we can see the same effect as we see when we add SEMA3F. Similarly, if we inhibit the Rho A partner ROCK, we get the same phenotype. The cells respond in a similar manner. Here is some biochemical evidence that indeed signaling through neuropilin-2 inhibits Rho A. So you see this signal has gone away by 30 minutes. And also relevant to smooth muscle function is phosphorylation of this protein called myosin light chain. And so whilst the total level of myosin light chain doesn't change, the phosphorylated, in other words, the activated form of that protein does decrease with SEMA3F treatment. And so we see this phenomenon in both bladder and colonic smooth muscle cells, consistent with, again, that tissue picture that I showed you. And then just one last piece of functional data from our cell biology studies. This is this bead twisting technique that I mentioned. When we add this protein SEMA3F, we see a dose-dependent decrease in stiffness, which is represented on the y-axis here. This compound, this is the ROCK inhibitor. So again, we're seeing a lot of consistent behaviors when we signal through neuropilin-2 with its naturally occurring ligand, SEMA3F, or with a chemical inhibitor of ROCK. And so just to summarize all of that, this is the pathway that we're seeing. When these proteins come together on the cell membrane, we're transmitting this signal into the cell that results in inactivation of Rho, inactivation of ROCK, and then some of its downstream targets, resulting in the favoring of this G-actin state, which is consistent with relaxation. So that's what we can do in vitro. I will tell you, if we artificially delete or inhibit neuropilin-2 in smooth muscle cells, we can see the opposite of this. So we see an increase in cell contraction. So this is all fitting very nicely. What about studying neuropilin-2 function in tissue? So here we've used two different types of mice, two different strains of mice. In the first strain, we have deleted neuropilin-2 using a variety of genetic techniques that we don't need to go into, but we can delete neuropilin-2 in all tissues. And so this particular mouse strain essentially never expresses neuropilin-2 at any stage in its life. And what we can notice using this muscle tissue tension testing technique, when we expose the tissue to electrical current, we can capture force generation. So in other words, a reflection of contractility that's represented on the y-axis here. So in the neuropilin-2 intact mouse, this is the response that we see. In a neuropilin-2 deficient mouse, which is shown in the black circles, we can see an increase in contractility. So this is in, without doing anything to the mouse, we just take the bladder out and we measure the contractility in response to a variety of stimuli. We can see an increase in contraction. We can also do this in a mouse strain where we delete neuropilin-2 only in smooth muscle. And we see something very similar. So again, electrical current on the x-axis and contraction on the y-axis. In the neuropilin-2 intact expressing mouse tissue from those animals, we see this response. In tissue from the mice with smooth muscle specific deletion of neuropilin-2, we see an elevated contractile response. And so this was very intriguing to us because there are very few strategies that allow one to enhance contractility. You heard about one strategy this morning from Dr. Rahman as it pertained to gut motility, but really there's been very little available in terms of either drugs or other strategies that have allowed us to increase contractility of bladder smooth muscle. So this was very compelling. But these mice are otherwise healthy. So what happens if we try this in the setting of a disease model? So first we had to develop a disease model to be able to test this potential hypothesis that restoring neuropilin-2 or knocking out neuropilin-2 would restore contractility. So how do we model altered bladder contractility in a mouse model? So we used a simple surgical technique, which is entirely artificial, but it does give us a model where we can capture increased contraction and decreased contraction. I'm going to just walk you through this. This is based on essentially occluding the urethra of the mouse. So you actually artificially create an obstruction and the bladder responds accordingly. I just want to show you, this is a picture of a mouse bladder in an animal that has not been obstructed. And you see the bladder here, it's relatively small, appropriately sized. This is what we see in an animal with a bladder outlet obstruction. You can see this enormous bladder, which is filled with urine. And so this is akin to an animal with urinary retention. So it is really striking. I mentioned this void spot assay in the beginning. And so this is what we see in the void spot assay, comparing sham animals to obstructed animals. And you can see immediately a difference in the pattern. So in the non-obstructed animal, you see large voids, relatively few of them. Whereas in the obstructed animal, you see many more, much smaller voids. And so this is consistent with bladder dysfunction, if you like. When we think about contractile function, this was our hypothesis. This is based on other studies in the literature where following a bladder outlet obstruction, at least, you initially see an increase in contractility. This is an adaptive response. This would be called compensated bladder function. And this is essentially the organ trying to overcome the increase in pressure that will occur when you obstruct the outlet. But over time, you can see the organ will start to decrease in contraction, decompensate, and very often come below the original level of contraction in the unobstructed organ. So we actually see this in our model. And again, this is looking in our tension testing setup where we can measure force of contraction on the y-axis, and this is time after obstruction on the x-axis. This is actually in response to a soluble factor that we can add to the organ bath, but we see similar outcome if we use the electrical stimulation as well. And what you can see is after 1 to 2 weeks of obstruction, the contraction is higher than baseline. This would be considered compensation, whereas when we get out to 4 and 6 weeks, it's dropping below the original level that we see in the sham animal. And so this is consistent with this decompensated state, so of reduced contractility. So with this model in hand, we set out to determine whether deleting neuropilin-2 using our genetic strategies could restore contractility of the bladder when we are out in this phase of the model. And so this is the setup. There's a lot of details on the slide, and really the only thing that you need to pay attention to, there are a couple of things. So we took the mice where we were able to knock out neuropilin-2 in smooth muscle only. The other feature of this model is that we can induce the knockout. So it's effectively like giving a drug to inhibit neuropilin-2 because we choose the time at which we can turn that gene off. So we can obstruct the animals or not. We waited for 4 weeks in this case to reach this purely contractile phase. Then we flipped the switch and we knocked out neuropilin-2 in one one of these groups, and it's this one in blue. The other 3 groups served as controls. We needed various controls for this experiment. We waited another 2 weeks, and then we tested smooth muscle contractile function using our tension testing setup. And so what we found was that if you just pay attention to the bar in blue, and again, this is measuring response to a particular contractile stimulus based on ATP. You can see we have neuropilin-2 expressed in 3 of these 4 groups. The one in which there is no neuropilin-2, in other words, that's the one that where we knock neuropilin-2 out, shows increased contractility, shown in this blue bar. Similarly, if we expose the tissue to the electrical stimulation, and again, we're measuring contraction on the y-axis, you can see that only mice with the deletion of neuropilin-2 show the increase in contractility. And so this suggests that we have identified a mechanism where we can actually restore contraction of bladder smooth muscle in the setting, in the face of an ongoing insult or injury. And this was really exciting to us. So this is all well and good. It's a mouse, who cares? Well, I can tell you that we were able to analyze some data from humans who had a similar outlet obstruction. And what we see, again, in our little schematic where we have compensation, decompensation, and we're interested in what's going on in this phase of the curve, when we actually look at that tissue, we see this inverse relationship between neuropilin-2 level and the contractile or compensated function. So this data set is from men with outlet obstruction, and we showed that those in whom bladder function was compensated had lower levels of neuropilin-2 than those in which their function had decompensated and were therefore poorly contractile. And so this is just a section of human bladder showing beautiful neuropilin-2 staining in muscle bundles. So what we're showing here, just to return to this really horrifying schematic one more time, when we delete neuropilin-2, what we're essentially doing is promoting this branch of signaling. So we are promoting contraction of the smooth muscle cell. We're essentially relieving the inhibition that would normally happen on this Rho-ROCK pathway. So we believe that mechanistically, it's the relief of inhibition of that Rho-ROCK pathway that is responsible for restoring smooth muscle contraction in the face of this particular insult. But we did want to look a little bit further to see if there were other hints as to how this mechanism was operating. And so we did a so-called discovery experiment using tissue from our global neuropilin-2 knockout as well as our smooth muscle specific knockout. And the details of this are largely unimportant, but what I want to highlight, so these red arrowheads in each case, just highlight proteins that are linked to cell structure and or regulation of smooth muscle contraction. I'll draw your attention to the line at the bottom. So in each case, we were able to capture the deletion of neuropilin-2 in our model. So that was very encouraging. But we have some interesting candidates here, including the circuit-2 pump. So this allows, it mediates calcium reuptake into the sarcoplasmic reticulum. Details, again, are not important, but it's a mechanism whereby muscle cells can continue to contract. And so we're excited by this candidate. We have other molecules related to calcium mobilization in the cell. Dr. Rahman mentioned calcium signaling earlier today. But suffice it to say, we have some interesting candidates that we can then pursue in our mouse models. So just to summarize, we've identified neuropilin-2 as a novel regulator of contractile activity in visceral smooth muscle, both in the bladder and in the gut. We've shown that if we inhibit neuropilin-2, and we're inhibiting particularly by deleting the gene in a mouse model, so this is entirely experimental, preclinical, but when we do that, we can actually increase and restore the contractility of smooth muscle in the face of an insult or injury. And at least based on our initial proteomics analysis, the knockout of neuropilin-2 is associated with changes in a number of contractility-associated proteins. So we're starting to build out the picture of how neuropilin-2 is regulating the contractile state of the cell. So I did note that we've just begun a collaboration with Dr. Musolino, and I think we would be very interested in testing whether indeed we can target neuropilin-2 in the ACTA2 R179H knock-in mice. We've also discussed the possibility of doing proteomics analysis on the bladders of such mice. And just to leave you with one last bit of information, clearly gene deletions are not feasible for translating these findings to the human situation. So we have begun to think about pharmacological approaches to targeting neuropilin-2. We have collaborated with a group at Cedars-Sinai Medical Center who've done structural analysis of neuropilin-2, so actually drill down to look at the molecule itself. And they identified a way that we could get drugs that would inhibit the function. So we wouldn't be knocking out the gene, we would be targeting the protein. And so we've begun some studies to look to see whether we could actually develop drugs against neuropilin-2. Again, we've just got some biochemical data. This is using a strategy. We've developed a simple bioassay in the lab where we can treat cells with SEMA3. We see a decrease in a particular protein, and so we're trying to use this as a bioassay to screen some pharmacologic agents. We have two. The panel here is just a repeat of this one, but essentially we're looking for agents that can reverse this decrease. So we have a signal, it goes down with SEMA3F, we're trying to bring it back. And so we have a signal with this particular agent, 4667, and we have signal with this particular agent, 8070. And so we have in hand a couple of lead compounds that we could then develop, use with additional expertise that we think could serve as the basis to develop inhibitors of neuropilin-2. I will say this work is probably a number of years away from ever coming to fruition, but it's certainly a starting point. So with that, I'm going to stop. I'd like to thank you for your attention. I know it's been a long day. Somebody I spoke to earlier said it's like drinking from a fire hose. So we'll turn the fire hose off. Thank you again for your time and your attention and for having me speak with you today.